Hello, everyone, to the second installment of this year's AIRR-C seminar. And this seminar is protein language model focused, which is really nice because it's a very hot field in the protein field in general, and also in the antibody field, and also TCR field. So in the air field. And so today we have Brian He from Stanford, and to briefly introduce Brian, he's an assistant professor of chemical engineering and data science at Stanford University, and an innovation investigator at ARC Institute, where he conducts research at the intersection of biology and machine learning. And he was previously a Stanford Science Fellow at Stanford University School of Medicine and a visiting researcher at Meta AI. And he completed his PhD at MIT and also was an undergraduate in Stanford. So he really likes Stanford. And so today he's going to talk about probably his latest paper, and his talk is entitled Learning to Read and Write Antibody Evolution. Very much looking forward. And then the second talk will be by Mamie Wang and from Yale, and she's also going to talk about language models and more benchmarking, I think, but also with respect to receptor specificity. But first, Brian. Yes.
Cool. Thanks, Victor, for the introduction. Yeah, I guess I have managed to stay in one place for a long time now, but it's great to be here. Yeah, my talk will focus on leveraging very large models that are trained on large amounts of data with large amounts of compute. But hopefully, when you do these sorts of things, you learn patterns. Over general, for example, protein sequences that can then be transferred to specific applications of interest, like the artificial evolution of human antibodies. Cool. Let's just get started. So just for some brief background, I'm sure all of you are aware of this. Evolution is a very powerful adversary. So for example, new variants of SARS-CoV-2, which are driven by evolution of the virus, cause these waves of disease and associated death. In a more extreme example, HIV can actually completely escape from the host immune system in just a matter of days, if not hours even. And more broadly, evolution is the driving force behind the worldwide emergence of multi-drug resistant bacteria, as well as drug resistant tumors as well. So if we want to be developing these better drugs or even better vaccines for these different diseases, then we're going to need better evolutionary models and better ways to understand and kind of counter the evolution of these diseases. So the approach that I and others have been taking to this problem is, rather than kind of lay out a set of evolutionary principles from just rather than you just laying out a set of evolutionary principles kind of from first principles, we're actually just going to learn directly evolutionary rules from data. So for example, there's hundreds of millions of protein sequences that are publicly available. And these are proteins that have been undergoing natural evolution. And so a question you might ask is, are there patterns? Are there non-random patterns in the sequence variation of these proteins that reveal something about the underlying evolutionary rules that were used to generate these protein sequences? So the idea of learning evolutionary rules directly from data is actually a very old one in biology. So for example, some of you might be aware of these amino acid substitution matrices like Blossom 62. So basically, based on different frequencies of amino acid substitutions, you can recover things like distance in the genetic code. So if things are closer in the genetic code, they're more likely to be swapped, two amino acids are more likely to be swapped out for each other, or biochemical similarity between different amino acids as well. So this is a very simple kind of evolutionary model. Somewhat more complex evolutionary models are based on sequence alignment. So you can take multiple related sequences, align them together, and you can then look at sites that are more conserved, sites that are more variable. And this might tell you something, again, about the evolutionary rules or the functional or selection constraints on a given family of proteins. And these models, based on sequence alignment, are actually very powerful. They're the basis of most modern phylogenetic methods, for example, but they do suffer from an underlying limitation. And so this is an illustration of this limitation that I like to use. I'm going to use a site-independent model to design a human name. And so if I look over the global human population for the most common first name in the global human population, that name is actually Muhammad. So this is the most common first name. Moving on to the middle name, considering this independently, the maximum likelihood middle name across the global human population is James. And then finally, moving on to the last name, the most common last name across the global human population is Wang. And so Muhammad James Wang is actually a very interesting name, but it's also a very rare name if you look over the global human population. And so what happened here, the idea is that the choice of the first name will influence the choice of the middle name and the choice of the last name. And likewise, in biology, for example, a protein sequence, the choice of an amino acid at the beginning of a sequence will influence the choice of amino acids in the middle and at the end of the sequence. And this idea that there are these complex interactions between sites in a protein sequence, and that the context of a sequence is important, is what's referred to in evolutionary biology jargon as epistasis. So this all brings us to the idea that Victor introduced at the beginning, which is the idea of a protein language model. And this one can think of as an evolutionary model that actually learns these epistatic patterns potentially from data. So protein language models are implemented with neural networks, and they're actually trained with very simple objectives. So for example, a class of protein language models called mass language models will take as input an amino acid sequence where a random subset during training is randomly masked or hidden from the model. So say 15% of the amino acids are masked. And then the model is asked to predict the value of those masked amino acids given the unmasked amino acids in the sequence context. And so if you force this model to do this kind of masked amino acid prediction task over hundreds of millions of sequences across all observed protein evolution, then you force this model to learn rules of how evolution has put amino acids together to form a protein sequence. And importantly, it's actually using that epistatic information. So it's using the value of some amino acids to predict the value of other amino acids. So we were intrigued by these language models as a way to both learn evolutionary rules and potentially even guide the artificial evolution of proteins in the laboratory. So in particular, we were very interested in this very simple kind of hypothesis or task. So sometimes the language model learns evolutionary rules in the form of likelihoods. So for example, if you blank out an amino acid at some position, the language model might say that the probability of an alanine appearing at the position is somewhat high, that the probability of an isoleucine is quite high, but the probability of tyrosine is so low you can basically forget about it, for example. So we ran a very simple experiment. So what happens if you just picked mutations to a protein that had higher language model likelihood than the likelihood assigned to the amino acid in the wild type sequence, and just use that as a way to kind of maybe narrow a pool for mutations that you want to screen in the lab for interesting properties like improving protein function, for example. So the specific task of interest that we applied this to initially was to the artificial evolution of antibodies. So in the immune system, all of you are probably aware of this, that antibodies undergo a natural evolutionary process via somatic hypermutation. And inspired by this, as antibodies have become a very attractive therapeutic modality, people also are interested in artificially evolving these antibodies to have higher affinity against therapeutic targets. And this is typically done via technologies like phage display or cell surface display, where you screen, sometimes millions of antibody variants for improved binding to the target of interest. So we were interested in using language models to maybe narrow that pool of variants from say, thousands or even millions of antibody sequences to around 10 or so. And so this is what we did, and I'll kind of walk you through the results on this slide. So we started with this antibody, MEDI8852. So MEDI8852 is a highly optimized antibody that binds to influenza hemagglutinin. And so MEDI had undergone both natural somatic hypermutation in a human, followed by additional affinity maturation in the lab. And so it's already starting off at a very optimized starting point. So in the first round of evolution, we use a general protein language model that's not specialized to antibody sequences, and we pick 13 amino acid substitutions to the variable region of this antibody. And these are the amino acid substitutions that have higher language model likelihood than wild type. So we test 13 of those language model recommended substitutions in the lab, which are these light blue dots in the slide. And we compare those to the binding affinity of the wild type variant, which is this gray dot. And to our surprise, we actually found that 2 of these variants actually improved affinity to hemagglutinin. And this was surprising because this language model had no idea that this antibody bound to anything, really, let alone to influenza hemagglutinin. And this language model is just trained on general proteins. It's not really specialized to antibodies and has no task-specific information or information about protein structure either. So we're doing antibody evolution. So in the second round of evolution, we combine these 2 affinity enhancing substitutions into a single variant, show that also improves affinity. And across a diverse panel of influenced hemagglutinins from different viral strains, we show sometimes up to a 7-fold improvement in affinity for this H7 version of hemagglutinin. And this is already starting off with a very highly optimized starting point. So we were surprised that we were able to achieve this. When we start off at a lower affinity starting point, so this is the unmutated common ancestor of MEDI8852, we test 8 language model recommended variants in the lab. 3, maybe 4 improve affinity. So in the second round of evolution, we make all possible combinations of those 4 affinity enhancing substitutions, sometimes observing additive and sometimes even synergistic effects on binding. Across diverse influenza hemagglutinins, again, this time we observe up to a 23-fold improvement in affinity because we're starting off at a lower affinity starting point, and there's kind of more room to grow from this kind of analysis. So we also show that this approach works across diverse antibodies that bind to diverse viral antigens, including the glycoprotein of Ebola virus and the spike protein of SARS-CoV-2. In all cases, we observe sometimes modest, but sometimes substantial improvements in affinity. In this one case, reaching this 2 orders of magnitude improvement for this Ebola virus antibody. And this is doing very, very lightweight directed evolution. So in contrast to a prevailing paradigm where you screen thousands or millions of variants, we're just testing 20 or few variants in the lab over just 2 rounds of evolution. And we really do think that the language model helps us narrow the search space for these mutations that at least are not functionally bad, or that the reason why we think these language models are working is that they actually potentially reduce this vast unproductive space of mutations, and thereby kind of indirectly enrich for mutations that improve, say, binding or some specific protein function. Just a brief slide. We also do this negative control where we show that these improvements in affinity are not just merely explained by polyspecific binding to proteins in general. So we use this assay where you basically test for antibody binding to soluble membrane protein via flow cytometry, and we observe no significant changes in polyspecific binding in any of our language model affinity matured variants. Again, just underscoring this point, but we really think the value of this kind of approach is kind of in this low N regime. So in contrast to traditional approaches in artificial evolution, or even in natural affinity maturation, which tests very large libraries of antibodies. Here, we're only operating in the tens of antibodies, really. And sometimes actually get comparable results to approaches that rely on larger libraries for screening. Something that is maybe a little bit beyond the scope of the community here today, but something I just briefly wanted to highlight also is that the methods that we use are not, the models that we use are not actually specialized to antibodies. They're actually general protein language models trained on, basically all observed protein evolution. And so in theory, the same algorithms can be applied to other proteins, other scaffolds, other definitions of fitness as well. And so in this preliminary work from the Alice Ting lab at Stanford, who we closely collaborate with, this student, Peter, showed that actually in this limited, again, in the tens of variants mutations to an enzyme, actually 2 of those improved catalytic activity of the enzyme. In one case, with this order of magnitude improvement, and this is using the exact same algorithms that we use to affinity mature antibodies. All I had to do for this really was replace the sequence of the antibody with the sequence of the enzyme, and it turns out the exact same process was able to enrich for mutations that improved catalytic activity as well. So everything I've been showing so far is with these sequence-only language models that learn properties of protein sequence alone, and they're already showing some promising results. So we can sometimes get pretty big improvements in affinity, sometimes more modest improvements in affinity. So then we were interested in, what if we could actually improve the model's ability to guide antibody evolution by adding structural information and actually adding information about the antigen of interest when we're guiding evolution. So how do we do this? So we actually use a class of language models that are called autoregressive language models, which are different from mass language models. So the way an autoregressive model works is it does what's called next token or next amino acid prediction. So given the first amino acid, the model will predict the second amino acid in the sequence. Given the first 2 amino acids, the model will predict the third amino acid. Given the first 3 amino acids, the model will predict the 4th amino acid, and so on and so forth. And so actually with an autoregressive model, you can, from left to right, basically sample amino acids in a sequence using kind of the likelihoods that are predicted by the model. So in addition to this autoregressive kind of model, we also provide the model with information about all of the backbone coordinates in, say, a protein structure. So as this model is kind of autoregressively generating a sequence, it's also being basically told that the sequence that it's generating needs to adopt the backbone structure that we provide to the model. And so this is often referred to as an inverse folding model because it's kind of the reverse of the protein folding or the structure prediction problem, where we actually provide the model with a full set of backbone coordinates, and then we ask it to generate a sequence that will kind of fold into or be compatible with that set of backbone coordinates. And so we're using inverse folding model from the MetaAI team that we trained for this task. And then we get out, something I wanted to highlight especially is that we're able to use inverse folding models to also score sequences similar to what we were doing for antibody kind of scoring. So inverse folding models also learn these likelihoods. And so an inverse folding model will say that, say if you have this T in this position here, the likelihood of seeing an L at this position here is 50%, so maybe pretty high. But if I mutate this T to an L, then maybe the probability of this L appearing in position 4 is now actually very low. So the way we can interpret this potentially is that the sequence QITL had high likelihood of adopting or being compatible with the structure that we provide to the model, but mutating the QILL could potentially be incompatible with this structure, is one of the reasons why there would be low likelihood of an amino acid, of this L amino acid appearing at this position. So you can basically think of this model as a way to score sequences that can be compatible with a given set of backbone coordinates. So if a mutation has very low likelihood under a given set of backbone coordinates, potentially that mutation might be destabilizing, or it might not even adopt that structure. It might adopt a different kind of stable structure. But if a sequence has high likelihood under a given structure, then that sequence is more likely to adopt the structure that you provide to it. So importantly, this task is not just limited to monomeric protein structures, but you can actually provide this model with the backbone coordinates of a multimer. So multiple chains that are in complex together, and then ask it to score mutations that actually stabilize or destabilize that entire protein complex. So this is some analysis done by a very talented student at Stanford, Varun. And he's kind of using these different models to score mutations to antibodies that, and he's benchmarking these models basically on their ability to predict which mutations lead to or kind of ablate binding affinity, in this case to H1 or H3 subtypes of influenza hemagglutinin. So if you, and this is done via deep mutational scanning experiments. So in this experiment, someone had made many mutations to an antibody sequence and then tested in relatively high throughput those mutations, the effects of those mutations on binding to these antigens. So if you actually take the likelihoods of a sequence-only language model or multiple sequence alignments of antibody sequences, the likelihoods under these models actually don't correlate very well at all with the actual experimental binding affinity of these various mutations when they're tested against these hemagglutinins. When we actually use an inverse folding model to score mutations to an antibody, the ability to, the correlation between the likelihood of those mutations and experimental binding affinities is actually better. And this is only when we're providing the model with the structure of the antibody. And this kind of makes sense. If you have a mutation that kind of destabilizes the antibody structure by itself, that antibody is also probably not likely to bind to something else. But then we can also provide the inverse folding model with the backbone coordinates of both the antibody and the antigen that it's binding to, and then score those mutations using the likelihoods under inverse folding. And when you do that, the correlation actually improves substantially. So for example, in this H1 setting, we have a 0.65 correlation. And we think that this is most likely caused by, the reason for this is because the model actually has information about the antibody and the actual antigen that it's binding to. So this is how we're able to add structural information both about the antibody and about the antigen of interest to this kind of language model guided evolution. And so we actually do this. So Varun then used these models to do the same kind of language model guided evolution that I was describing in the earlier slides. But an important thing that he's doing is now he's directly screening these antibodies, not using binding affinity, but actually using neutralization potency against different pseudovirus. So this assay will basically test the ability for these antibodies to prevent viral infection kind of in vitro. So Varun is, again, scoring these different single amino acid substitutions to these antibodies in the first round of evolution, and then using the likelihoods assigned to the combinations of those mutations in the second round of evolution. And actually, for this antibody, this is Li-1404, this is bebtelovimab. It's an antibody that was clinically approved under emergency use for use against COVID, but it had lost efficacy against the Omicron variant. Varun was able to actually restore a lot of that neutralization potency back using inverse folding. And this is neutralization potency against the BQ 0.1.1 Omicron kind of subvariant. And then again, for this other antibody, SA58, which was it was not clinically used, but also initially had very broad neutralization potency against SARS-2 viruses, but then kind of lost that efficacy again against Omicron. Here he was able to almost completely restore, or over a couple orders of magnitude actually restore neutralization potency to this to the BQ.1.1 subvariant of Omicron. A very interesting finding is actually that a lot of the mutations, both in the protein language model and in the inverse folding model guided antibody evolution, are actually structurally distant from the antibody paratope, or the actual binding interaction to the antigen. So this is an important point, but actually when we do this language model guided evolution to the antibody, we don't just restrict the mutations to the CDRs, we actually allow the models to design the entire variable region of the antibody. And so there's many mutations actually that do confer increased affinity or increased neutralization potency or increased breadth that are in the framework regions of the antibodies. And it's also known that actually natural affinity maturation will very often actually reach into the frameworks for affinity improving mutations as well. So this is another kind of thing to highlight as a key feature of this method. And finally, just to put this in context, so within the kind of literature of machine learning guided evolution, there's this kind of literature where you can train on task-specific data, and you train a supervised model to guide antibody evolution, and then use the outputs of that supervised model to guide additional rounds of evolution. And we're kind of operating in this regime where we actually assume no task-specific training data. So the inverse folding models that we use, they were trained on very large corpuses of protein sequences and both experimental and predicted protein structures. But actually for the everyday researcher, when you go and use this kind of approach kind of in your lab, for example, you actually are not required to provide the model with any task-specific information on your antibody of interest. And the model is still able to improve for this one antibody around 50% or so, that the neutralization potency in around 50% of the variants that it suggests. And for this other antibody, around 80% of the variants that it suggests. So we think this could be very valuable in areas where you might have some task of interest, but you have no or very little task-specific data with which to train a large supervised model. In this case, you can actually use a pre-trained model directly, and it can be helpful for whatever task of interest you're interested in. And then potentially, the data that you collect from the initial round that's guided by the unsupervised model can be used in future rounds of evolution as task-specific training data too. Okay, so this concludes the kind of the research that I wanted to talk about. So something looking forward, and we kind of we're kind of moving towards this direction with this paper, where we show that we can basically co-evolve the antibody with the antigen. But something I'm very interested in is potentially using better models of how antibodies and antigens bind to each other to maybe recapitulate some of the coevolutionary trajectories that we see in natural evolution. So for example, and this is data from HIV, in HIV actually there's an arms race within a single host between affinity maturation via somatic hypermutation in the immune system and the actual within host evolution of the HIV virus. And so a lot of the reason why HIV is, why we haven't, for example, developed an HIV vaccine is that the immune system is currently losing this arms race. But if you had a better ability to kind of simulate this coevolutionary trajectory, then potentially you might be able to find ways to kind of prime the immune system so that it might not lose the arms race, or confer maybe better protection for longer than anything we have right now in terms of like an HIV vaccine. Or even looking not so far ahead, evaluating antibody therapeutics based on their ability to resist evolution of a virus, even though multiple antibody therapeutics might appear to work equally well right now, potentially some antibody therapeutics might be more resistant to future evolution than others, even though they appear to work equally well in the present. Okay, so here, just for some brief summary, we discussed these language models of protein sequence that just learn the statistical rules of protein sequence variation alone over these big corpuses of protein sequence data. So these language models are just learning the distribution of a protein sequence. We also have these inverse folding models that learn the probability of protein sequence given structure. And in both of these cases, we can use them to affinity mature antibodies and guide antibody evolution in an unsupervised way. The benefit of the protein language model is that it just requires the sequence of the antibody alone. You don't even need to provide it with any information about the antigen, and it seems to, at least in many cases, improve or be able to enrich for mutations that improve function. For the inverse folding models, we can get better performance in the antibody than the protein language model, but this does require, say, the three-dimensional structure of the antibody antigen complex. Very briefly, this work was not done by me alone. It's done with a very talented network of co-authors and collaborators. So I basically wanted to acknowledge them here, in particular Varun, who did pretty much all of the work and for the inverse folding model and validations. And finally, thank you all for your attention.
Thanks so much. This was really fantastic, Brian. I'm sure there are many questions, and if you have a question, please raise your hand. The first one, maybe I can have the first question. You talked about epistasis in the beginning, and that language models capture this epistasis, but to what extent do we know that this is true, and to what extent do we know what epistatic rules they capture and what they don't? So what is the ground truth against which you are comparing? Yeah.
Yeah. So okay, so I think we've actually looked at this a lot. So I mean, fundamentally, the models are epistatic models in that they actually do, like in terms of their pre-training task, they take the information from some amino acids and they use that information to predict the value of other amino acids. So almost like by definition, they need to learn epistatic information. What we've shown
Maybe to
actually
refine, yeah, maybe just to refine my question, to what extent do we know that they learn not noise rules, but the actual biological? So I would really like to understand do we have biological ground truth? And then can we compare to what extent do they learn and to what extent don't they learn a given epistatic landscape, basically? Yeah.
Yeah. So we actually, I guess this then becomes part, like, how do you define epistasis, I guess? If it's just any kind of statistical dependence, or if you needs to be sensitive to actually the biological mechanism by which mutations are dependent on each other. We've actually done a lot of work trying to see if these language models were, so a lot of people have done these mutations in protein evolution will actually show epistasis among pairs of amino acids or something. So where, for example, the combined effect of two mutations is different than the additive effect of the individual mutation. And actually, in many of these cases, the protein language models that we use just don't learn this information, or appear not to be sensitive to this information. So what we think is happening is maybe they're learning like a very blurry picture of epistasis that that takes into account like a lot of information across the protein sequence. But in terms of actual mechanism, like a biophysical mechanism, or even mechanism agnostic, just being sensitive to mutations that like pairs of mutations, like having a very kind of high resolution sharp picture of epistasis. That's like not really learned by the models right now. And you're right that maybe encoding kind of biophysical information to these models could make them more sensitive to these kinds of like precise changes in epistasis. But on the other side, I think, I mean, it's been shown by many groups actually that at least in certain tasks where you try to predict the value of mutations and have their and their effects on different notions of function, so people will benchmark these models basically on their likelihoods ability to predict experimental measurements from deep mutational scanning. The protein language models that do learn statistical dependencies among amino acids, which some might define as used to define epistasis, the models do outcompete, I guess, or outperform models that don't have any epistat or don't have access to any epistatic information or statistical dependencies. So in that case, I think it's definitely helpful in making predictions, but I think there's still, I mean, maybe a lot of work if you want kind of a high resolution picture of epistasis, and maybe actually potentially the models that do have some information about structure, or that are refined with actual biophysical or biochemical experiments, could be directly steered in a way that they learn epistasis. But this is ongoing research. Yeah, it's a good question.
Great, thank you. Felix and then Tim.
Well, given that discussion, I'm not sure if this question makes sense, but with your emphasis on epistasis, and you discussed additive versus non-additive improvements in binding. But I mean, with strong epistasis, you'd expect some combinations to be negative. I mean, did you see any sort of, were you just kind of reporting the best binders, or did you in fact not see any combinations that were actually wiped out? Or yeah, okay.
I don't know. So it was interesting. We did a little bit of work on like seeing not very thorough systematic work, but just like looking at which mutations kind of that in combination the model thought were better, and that when we tested in the lab actually worked better. So actually for the protein language model and to some extent the inverse folding model, the models, and this maybe indicates that the models are limited in their ability to reason about epistasis. But if the model thinks that an individual mutation is beneficial or has higher likelihood, and there's multiple of such mutations, and you just combine them and give it to the model will also just think that the combination is better. And this is kind of related to how these models are trained potentially. But actually when we test these mutations in the lab, we actually for inverse folding especially, and this is in a very low end setting, so I know this is again not very systematic, but we also observe no negative epistasis actually when we combine the mutations. So I mean it's in many cases not additive. So like if two mutations were beneficial in the first round, their effects on neutralization potency or binding would be beneficial together in the second round, and then but the effect would not be additive. So but sometimes it would be additive, sometimes it's like synergistic, and as was the case here. So I don't think, yeah, it's we haven't seen like a very one-to-one kind of mapping between how well a language model thinks individual mutations work well, and then if you combine them, the effect on language model likelihood typically just goes up anyway. So the combinations kind of need to be tested manually. But yeah, I think it's a really interesting area of research because ideally, if we wanted a good model of protein evolution, it would be sensitive to these small changes and would not require like experimental feedback or whatever to kind of recalibrate things on each round of evolution. But it's productive enough where like if it's productive enough where actually if you have mutations that work well in the first round, we're like pretty confident actually that like they'll work in combination in the second round. This is also something our collaborators who work on enzyme evolution have seen. There's actually no negative epistasis for the language modeling mutations on catalytic activity there as well, which is kind of interesting. But
yeah, it's interesting. Yeah.
But I don't have data to make any broad statements. I guess Tim is this.
Tim, yeah, Tim.
Thanks. Yes, super interesting work. I was wondering with just the language model improvements, like what structurally do you think is happening to drive those improvements? And like, is the language model thinking about the likely antigen specificity, you think, of these antibodies, or is it kind of making it more thermostable or something like that?
So yeah, it's interesting to speculate. What we did observe in our experiments, we actually did test the thermostability of the antibodies, them's like the fab of the antibodies with like just like a thermal melt. And actually we observe kind of similar things with binding affinity. So there's not a consistent relationship between language model likelihood and stability. So in like some of the cases, the melting temperature would improve. Some of the cases, the melting temperature would decrease. And it was almost, there's one or a couple of particular cases, I think for Medi, where actually there was like an 8°C drop in melting temperature of the fab, but then like the affinity of the antibody improved like 20-fold or something, which was interesting. In terms of what I think and what I can speculate about what the language models are doing, they do learn an image of protein structure. This is a well-known fact. Like you can use language models to predict protein structure pretty well, actually. So potentially when it sees things from like an immunoglobulin-like fold or something, it might know certain regions. I mean, it does have some natural antibody sequences in its training data. So it might use that information as well. And kind of the inherent variation within the human antibody sequences in its training data, combined with its more general information about protein structure, maybe guide evolution to regions of the antibody that might okay, that might improve. The reason why I'm like struggling to say things is because like this is total speculation. So maybe I'll just punt on the question.
I'm wondering like like if you mutated the CDRs, like I think some of your mutations are in like framework regions, right, that don't impact the antigen. But I'm wondering like would those same mutations have been suggested for like same germline origin of the antibodies, but like different CDR regions and like different antigen specificity?
Yeah, I mean, so one thing, especially in the language modeling case, is that if you were trying to evolve the antibody for a different function of interest, the language model would recommend the exact same variants, like by definition, because it's function agnostic. So this will probably typically work when if you think of these models just as a way of kind of productively exploring a local fitness landscape and reducing just clearly bad substitutions, then this using language models as a method for guiding directed evolution will only work if there's a little bit of a function already in place. So for example, if you started off with an antibody that didn't bind to anything, or you were trying to evolve an antibody towards some function that's very different from the function that's already in place. So you start off with an antibody that is meant to bind influenza, but then you want to turn it into a catalytic antibody or something. Like that would be very hard or impossible to do with this. So I think the value with this kind of method is if you have like a little bit of a function in place and you want to kind of diversify that antibody kind of around the local sequence landscape and remove a lot of just clearly bad areas. And when you do so, you naturally kind of indirectly enrich for mutations in the local sequence landscape that improve function, then it's a valuable strategy. But if you're trying to engineer some de novo function, this is probably not gonna work.
Yeah, I have more questions since we have also more time, which is good. Yeah, so this slide is nice, but also the other ones where you then tested the antibodies and some were better and some were worse, etc. Can you briefly explain how you picked which antibody variants then you actually tested in the lab? Because there must have been many more that could have been also been good candidates, I guess, or yeah.
Yeah.
And maybe expand a bit on what do you think would have been the underlying diversity of many other good variants, maybe. Yeah.
So in terms of the numbers, like the absolute numbers were just limited by basically the experiments we use to do evolution.
Yeah.
So for example, if you're screening with just recombinantly purifying the antibody by hand and then manually testing it for binding, like the BLI that we were using, or pseudovirus neutralization, and it's just one person, me or Varun, doing the experiments, then that limits the n to around 10 for each antibody. So that's, I mean, it's like a very practical consideration in terms of like how we pick the actual n, and that basically guided the various cutoffs that we use to pick mutations. So for example, in the antibody in the language modeling work, we actually had a little bit of this kind of Baroque scheme where because we had an ensemble of language models. So we had 6 different language models actually. And some language models would say that certain mutations were higher likelihood than others, but then another language model wouldn't think that it's higher likelihood. And we actually found that if you take the consensus over multiple language models, it typically works better at recommending mutations that improve some function of interest. So we kind of have this consensus scheme where we say like we need at least 2 or 3 recommending language models, and then based on that we use that to set the cutoff to be around 10 or so in the first round, because here we're just using a single for inverse folding actually. So actually for the language models, if you just use the cutoff of higher language model likelihood than wild type, you'll get around like 10 variants for antibody just naturally, actually. For inverse folding, what we found was when we use that cutoff, we still get a ton of mutations that have higher language model likelihood than wild type, which is interesting maybe by itself. So we needed just another way to filter. So we just take the top, and I think it's 20 or something actually in the first round here. So and you had a second part of the question. Should I?
Yeah, what's the but maybe you answered it. What's the underlying diversity? So what's the total diversity of good variants? You expressed 10, but could there have been 10,000 or something that would have been also good? Yeah.
Yeah, it's interesting.
Yeah.
You're doing like kind of a traditional directed evolution approach where you in the first round you only pick single mutations.
Yeah.
Then you make combinations in the second round.
Mm.
So like your total space is limited by the sequence length times 19 or something. Yeah, it's interesting. We have kind of done these experiments where people had done a deep mutational scan, and we say if we take the top n, I think we have these results both in the inverse folding paper and the language modeling paper. But if you take the top 10, what's your like success rate? If you take the top 50, what's your success rate? Actually, I can't recall the results off the top of my head, but they were you can go down pretty far, like to the top 100 or something, and still get like significantly higher enrichment for function than just random. But once you get down to, I mean, just large amounts of the sequence landscape in terms of single mutations, then it just naturally approaches random anyway.
Yeah.
And then in terms of diversity, it's all just at one amino acid change away. So it's yeah, they're all equally diverse. I guess it's one substitution from wild type in terms of sequence diversity. Yeah.
Okay. Yeah. But yeah, I see. So there are no double mutant, double combinations, basically.
Exactly. In what we were doing. So, actually like when we were doing the language modeling work, I was skeptical that even the single mutations would express or even like maintain stability. So we were very conservative. In hindsight, but hindsight is always, I guess, 2020. We could have been a lot less conservative and actually introduced a ton of mutations in the first round guided by the language model. But yeah, so I think that's really interesting for future work. Like if you had a combinatorial library guided by the language model and kind of control.
Because we know from all these trastuzumab deep mutational scanning experiments that have been published, right, that there are many trastuzumab variants that are not one away, but 2, 3, 4, 5 away from that do express and that bind in a similar affinity, maybe a bit lower, a bit higher than trastuzumab. So the potential binding space is actually quite large, which is interesting. So, but and maybe then the last question from my side, with on your future perspective slide. So how do you, maybe you don't want to say, but how do you experimentally or with protein language models or both want to model the coevolution of antibody and virus? So then you have data from virus evolution and the antibody evolution from mice, or how do you see that? Yeah.
Yeah, I think I mean I have some ideas, but we don't have any like concrete, I guess, results or things. I mean there's different ways you can take this. Yeah, like one way would just be if you actually had coevolutionary data, and there are actually a lot of coevolutionary lineages for example HIV. I mean it's not in the thousands, people do in the tens, which you can potentially use to as validation, not as training. Like if it's sufficiently, like if you set up the experiment correctly and use it as held out validation.
I think
There's multiple approaches. If you just had a better predictor of binding affinity, that would get you along a lot of the way there, I think. But I mean, and then like affinity might not be a perfect predictor of like neutralization. There could be some avidity effects. There might be other effects. I know for example, in the T cell world, like sometimes higher affinity does not always lead to like better activity actually. So yeah, I mean, this is definitely aspirational. There's a lot of work to do for something like this, but yeah, I think just even in a more simple problem, like how do we reliably rescue binding to escape variants or something? Like I think we have like pretty good progress here, potentially make better progress, make this more reliable. Like that would be useful without having to go like multiple rounds of evolution in the future.
Great. Are there any last questions for Brian? Otherwise, thanks again a lot. This was really, really nice. And then we come back in 4 minutes for Mimi's talk.